partially today. So if you last time when we were together talking about fungus, I think we left off with these guys here. This is the small group of fungus, the glomeromycetes. Okay, that little tiny group with 160 species. But again, it goes to show you that good things come in little packages, right? Because with only 160 species, these are the guys that form those mycorrhizae. Remember, we've been talking about those a million different times. Mycorrhizae are those little extensions of the root system of a plant, really. They're not from the plant, though. They're fungus, right? They're these little threads here, and what they do is they increase the surface area. And glomeromycetes, nearly all of them form this kind of mycorrhizae. Remember, the one you saw in class was, okay, so what they did is they took a root of a cell. So like if this was the root of a cell, or the root of a plant, they sectioned it, chop, chop. So like a hamburger patty, and then they laid that down flat on your slide. So what you're looking at is the xylem and the phloem and all that vascular tissue, right? So you're looking down through those tubes. And remember in your slides on the outside, oh, I can do it. Look at this. Check this out. I can make it blue. On the outside, there was that bluish green wrapping. And if you focused in close enough, right, if you went up in magnification and you played with your fine focus well enough, you noticed that it looked kind of the way I just drew it, right? It was kind of fibrous. That was the mycorrhizae that you look at. Now, that's not this kind. That's an arpuscule right there. Kind of a horrible name, right? Arpuscule. The kind you were looking at is called ectomycorrhizae. Still have to look at how to spell that word sometimes. Ectomycorrhizae. Ecto, right, because it's on the outside. So two types of mycorrhizae. Don't confuse the two, because, like I said, the one you looked at in class or the one you looked at in lab is not the one that I'm talking about here. There are different kinds of mycorrhizae. These guys aren't the only ones who make these glomeromycetes. They're not the only ones that make these mycorrhizae. This is the type of mycorrhizae that is the most prominent, though. See, 80%. So even once again, a small group, but a very important group. So now let's move on back to a bigger group. This is actually the biggest group. 75% of fungi out there belong to a group that we call the Ascomycota. Now asco means up. So these are what are called sometimes as the cup or the sac fungi, right? And there's one down below in the lower left, which I neglected to put the species name in there, so I have no idea what it is off the top of my head. But that looks like what I'm talking about, right? It looks like a cup. This thing here, Morchella, that's a moral, M-O, not A-L, but M-O-R-E-L. And even though it kind of looks like I was going to say a tadpole. No, not a tadpole, Steve. A toadstool. Even though it kind of looks like a club fungus that we were talking about in class the other day, the cups are these little pits. See? So it's still a cup fungus. This is another one, the tuber melanosporum. That's another species of an ascomycete. The cups are just a lot smaller. These are sometimes called, or well, commonly known or common name is truffles. Truffles, right? This is another sac fungus, displaying exactly what I'm talking about. Speaking of truffles, do you know why they use female pigs to find them? Truffles are very valuable. Look at that. What is this? I can't with my glove. 8 ounces of black winter, flash frozen black winter truffle. 8 ounces, 500, and is that $73.99? $574? Holy cow. Has anybody ever ate a truffle? I understand that, like, you get it, like they shave it on stuff, or like I heard someone tell me once it's like an oil that they flavor things with. Gosh, I would. Yeah, that's my dad. I don't eat toadstools, he always said. I like mushrooms. Some I don't care for, or care for some other more than others. But man, I tell you, I would love to know if a mushroom could taste so. What's it taste like? It's like caviar, right? First time I tried caviar one time, I was at some party that I was at some party I probably shouldn't have been at, for the kind of person I am, but they had caviar, and it's like BB's covered in fish oil. It was disgusting. Well, one of these days, I'll have to try a truffle just to see, but I'm not paying $574. Even if you bought, I don't know how much, like a pinch Can I get a pinch? No. All right. Okay. Now, bread yeast is also an ascomycete, although in your lab manual it calls it a deuteromycete. Remember, that's because deuteromycete is that collect-all group that if we don't know their sexual stage, and yeast, that's kind of still being debated. Okay. Molds, the stuff that grows in your bathroom walls, truffles, morels that we talked about, lichens. About a quarter of the ascomycete species, about a quarter of them exist in this weird sort of, well, not sort of, but obligate symbiotic relationship, right? I'll go back to the pigs in just a minute. I just remembered I just remembered that I asked that question, forgot to answer it. But lichens are what we call an obligate, there you go, Steve, an obligate symbiotic relationship. They have to live together, even though they're two different species. I mean, one's a fungus, you know, so that's a completely, that's a kingdom fungi, and the other one is either a plant kind of protozoan, the green algae, or holy cow, you could even be a prokaryote. You could be a cyanobacterium. And these things have lived together so long that later on we'll take a look at them. They have species names for lichens, which totally screws up our whole definition of species at the beginning of the semester, doesn't it? Because this isn't one organism. This is two organisms. That's that stuff you see on the trees. We've talked about, we took out the nature trail. We've talked about lichens. I showed you the kind in class too. We'll look at them again later on. But those are those splotches of will look like paint up against the tree. Those are lichens. All right, back to the pig thing. Why do they use female pigs to find truffles? Truffles grow under leaf litter. So the pigs find them, and then the dude with the lead says, no, you're not eating that at $575. But why did they use the female pigs? Anybody know? Yeah, exactly. And there's a hint in the picture, right? Female pigs are sniffing out the, does anybody know what a pheromone is? Dustin, you know what a pheromone is? Sounds like hormone, so it's kind of like that. Yeah, there you go. Pheromones, I like to think of pheromones as being sort of external hormones. They're scents or molecules or like Dustin, all of that. That's smell, what Dustin was saying. Smell that's given off. Sometimes in very small quantities, but they many times they signal things like, say, sexual receptivity. Like, for example, they did this research one time. This is how incredibly sensitive certain species can be to smell. It was on a moth. I don't recall the species, but they found out that a male moth could sense the female's sexual pheromone. Now, not all pheromones are sexual. Like ants, when they leave the little trail, they leave a little bit of chemical. That's how they're like. Oh gosh, brother and sister wandered in the woods, lost. Hansel and Gretel. They're like Hansel and Gretel. They're dropping little chemical things on the ground. That could be a pheromone as well. But this male moth, had such an incredibly sensitive sense of smell that it could sense the female moth. Now, this is a moth, okay? Female moth's pheromone a mile upwind. So the moth is against the wind. The male is against the wind a mile. It can still smell. There's a female moth around here who's, in heat? No, not in heat, but sexually receptive. Yeah, no kidding, right? There's some debate or thought about whether humans have pheromones. We might produce pheromones, even maybe sexual pheromones, but I wonder sometimes if our sense of smell, has diminished to a point where we really don't. We don't have a really great sense of smell. Sometimes you can even see they sell pheromones like female or male pheromones. You can put them on yourself and then, I don't know. I guess everybody just can't stay away from you. But yeah, the female pigs are going after the male pheromones, and then of course when they see the truffle, they're like, "Oh, something to eat," and that's where the guy comes in and says, "No." Okay, let's take a look at how these sac fungi reproduce, and again, I told you the other day that fungi are all classified; they're all related based on on the way they reproduce. So let's do the simple one first. Many fungi, all fungi really I hate that word all. Most all fungi, or the fungi that I know, have asexual reproduction. And that's very simple, that a spore germinates and turns into mycelium, and the process is repeated like you see in the upper left. Now I want to make sure that we don't have any confusion here, but there are two types of spores that are created. The one I just mentioned to you, the ones that are produced asexually, the asexual spores are called conidia or conidiospores. And conidiophores are structures that produce these conidia. Just like the sporangiophore, remember in the zygote in the black bread mold, had that lollipop thing on top? We called that stalk the sporangiophore, the structure that creates the sporangium. Now, you don't have a sporangium in the conidiophore, but the conidiophore is still going to be able to produce those spores, but it's doing it asexually, so it's just dividing. Okay, so there's the asexual spores. Now, ascomycetes or club fungi, or sac fungi, also have a sexually reproductive. So let's start with that weird part that fungi have, the part in the purple. The part, you remember that I was talking the other day about, technically all cells fuse together, or plasmogamy, and then the nuclei fuse together, karyogamy. It makes sense. You couldn't have one before the other. Now, in animals, you and I, that just kind of happens to happen pretty quickly. Once the sperm penetrates the egg, boom, 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 it fertilizes and moving on to a zygote. But what the fungi have done is this is their really, really cool evolutionary innovation. They play with that stage in between. They play with that stage in between when the cells fuse and when the nuclei fuse. Okay, so that's called that heterokaryotic stage. So if we back up just 1 second from plasmogamy, if a conidia were to, it's sort of like the way pollen sticks to the end of the style on a plant. The stigma. It's kind of the same way, probably with pheromones, right? They're smelling each other out because these guys got pheromones too. So, a spore would attach to the end of the hyphae of another mating type, right? We don't have sexes. We don't have males and females. We have mating types. Okay, now, so you follow in the blue and the red color, those are the nuclei. Now, plasmogamy is going to occur, but you're not going to fuse the nuclei yet. You're in that purple range. So you're going to form something called an ascus. So when the terminology is all kept rather consistent. So ascomycota produce an ascus. Now, you were looking at an ascus, let me erase some of my scribbling, the other day in lab when you were looking at Peziza, okay? Peziza was the one that when you looked at it under the 4X, it was stained sort of a blue-green color, and it was, oh, come on, pen. And it was shaped kind of like that, right? And then when you zoomed in on it a little bit closer, you notice it was a bunch of little finger projection, wasn't it? And if you looked inside there, you could see that there were things that were stained kind of a purple color in some cases. Even if they weren't stained, you could see there was something inside these little fingers. And I told you those were spores, okay? All right, those things that I'm talking about there, a singular, is an ascus. All right, now if you notice, I'm going to jump over to the other side of this cycle for just a second. But in the lower left, a collection of ascus, more than one, are asci. So you were looking in Peziza, you were looking at the asci. And inside of those ascus, or inside those asci, there were spores, specifically ascospores. See how we're kind of, and that's all ascomycota. Now, the thing that produces the spores, like the actual fungi itself, all right, is known as an ascocarp. A carp is the fruiting body. What we mean by fruiting body is we're not making fruit here, obviously, but we're talking about the reproductive part, the sexual reproductive part. Right? See how that all works together? Now, the spores that are produced by the ascocarp, see, ascus means sac, okay? Those spores, like these right here, those are sexual spores. Don't confuse those with the asexual ones. So these ones down here, these are not conidiospores. Those are just spores, ascospores to be precise. All right, let's go back over to that middle ground again. So we form these dikaryotic hyphae. I want to make sure that you understand the evolutionary significance of that. Now, what that provides, or with the advantage that that potentially provides to these organisms, is being dikaryotic, in other words, not fusing the nuclei, is you have the benefit of all of the genetic information. So you don't get the dominant and recessive thing in some cases that you might get in the nuclei when they fuse together. Also, the longer the fungi stay in this dikaryotic stage, the more times those chromosomes are going to get together and engage what's called genetic recombination. Now, genetic recombination, folks, is anytime you rearrange the genetics. Remember how viruses, remember I told you viruses can inject a little piece of DNA into a bacteria? That's genetic recombination. When the sperm and the egg fuse together, that's genetic recombination. When crossing over occurs in prophase 1, that's genetic recombination. So that's what I mean by that. In genetic recombination, what does genetic recombination result in? What do you get from doing that over and over and over? You know, think back to meiosis. Why do you do crossing over? Why do your cells, I guess I should say, why do they do crossing over? Yeah, it's all about that, right? I must have said it a million times, haven't I? So we get variability there, and then look what happens, because we're not done. We're not done shuffling the cards yet. The more variability, the better. If it goes on, or when it goes on to karyogamy, and that's when the nuclei fuse. That's where you get a diploid nucleus, a zygote. Now, here's one of the big differences between fungi and animals, right? You're the result of mitosis in a zygote. It's a very clinical way of saying it. But what happened is when egg and sperm fertilized to make you, after that you just kept adding cells through mitosis. Mitosis got bigger and bigger and bigger. So here's a big difference with fungi. They don't do that. They fuse together the nuclei and immediately go back into meiosis. So they use the zygote stage, that short-lived diploid stage, the one in which we've chosen as animals to kind of make the most of your organism. They've chosen to simply use that to turn around and go through meiosis, making more variability again, right? Meiosis is all about variability. So you have all these different kind of haploid nuclei, which then divide by mitosis a lot. So you have all these different kinds of, I should have highlighted the ASEO there in red, right? The ascospores. Variability, variability, variability. You get a bunch of those ascus or an asci all together, and boom, you got yourself a boatload of spores. And they're all different. So the thing I've been saying from the very beginning on every organism we've gone through over and over, that's why, again, right, I spent so much time at the beginning on evolution. That's what drives all this stuff. If you're wondering what makes life so diverse, that's what it is. Organisms are constantly trying different things and finding new ways to survive. It's actually kind of amazing when you think about it, right? Any questions on the sac fungi? The sac fungi. Who's come in since I started? Hi, Kaylee. How are you? Dustin? Rayanne, how are you today? All right, let's take a look at these guys. These are what normally we call the club fungi. Now, that's not because it's the toadstools. I told you that in lab the other day. This is that one, isn't that the one that the Cheshire Cat or the grinning cat in those Alice in Wonderland stories? Isn't that what it was sitting on, one of those kind of mushrooms? Yeah, I think so. Lewis Carroll, the guy who wrote those books, what a strange individual. I think he was an opium addict, just like Poe, Edgar Allan Poe. Yeah. Now, Basidio does mean club, but again, it's not because that it's necessarily looks like a toadstool. I showed you that puffball in class with all those spores. You know, that's a club fungi. Shelf fungi, like the turkey tail that I've showed you. These really cool ones I made in Vail. I'd love to see something. Wouldn't that be neat? I mean, come on. You're walking through the woods and you're going, what? This is cool. Look at that. I would almost think it's like some insect nest or netting kind of arrangement, like a spider. This is smut. I know. I guess maybe that's why pornography is called as a slang as smut, because it's kind of, well, to some people, whatever, disgusting, right? And these are rusts. Rust looks like rust. These are all examples of a club fungi, but or basidiomycetes. Now notice, like I said, they're not all shaped like a club, even though the ones we typically think about, the toadstools there, they are. There's a reason why they're called basidiomycetes. There's a reason why we're called club fungi, and it has to do with the same reason why the ascomycetes were called sac fungi. There's a club in here somewhere. Let me show you where that is. But these are important wood decomposers. Some of them species are mycorrhizae, pathogens, right? Rusts and smuts. Now, it's a weird thing, but that smut, that particular kind of smut that's on the corn, that's not good for the corn. But later on, I talk about it where, one man's food poison is another man's food. That apparently, in some cuisines, in some Hispanic cuisines, that smut there is a delicacy. I've never had it, but I mean, I would try it. I mean, I eat other mushrooms, so I don't know. I'd try it. It sounds kind of interesting. Oh, okay. This was not Peziza, but this was Coprinus. And when you, the other day in the lab, and when you looked at it under 4X, this was the thing that was like the circular rosettes, maybe, if you understand what I'm, like this. You remember that? It looked like that when it was under the low magnification. All right, so in that case, what they did to make that slide is they took a toadstool, like I'm drawing up top, and they sectioned it this way. And then they laid that sliver down on a slide, okay? So what you were looking at on the edges there were actually the gills of, like in those typical toadstool. So this is a picture that should look familiar to you, right? That's what you were looking at the other day underneath the microscope. So the blue part here, that's the edge of the gill, and those little lumps that are sticking out. Now, this is where it gets its name, basidiomycetes. The clubs that we're talking about, is not that many of them are toadstools, but a basidia, these little blue sort of protuberances that you see right there, that looks like a club. That's where they get their name. That's the basidia. Down below, basidia is plural. Down below, you can see a basidium. See the basidium down below? So it looks like a club. The sterigmata, cool word. The sterigmata is really nothing more than that little sliver piece right there. That part that holds the basidiospore, right? So the basidiospores had sort of a brownish color to them. The basidia were kind of hard to see. The basidiospores were much easier to see, but again, it's the basidia that give it the name, the club fungi. Now, these organisms do asexually reproduce, but you notice it's not on the screen here, and that's because they typically do not. So they do, that's why I said they do have asexual reproduction, but we're going to actually just look at the sexual part because this is typically how they do it. So your average, everyday, run-of-the-mill, toadstool there that you eat and see out in the yard is going to reproduce sexually. All right, so let's go ahead and start right about here. Two mating types come together. Remember, we don't have sexes in fungi. There's not girl fungi and boy fungi. There's two different mating types. Fused together by the cells, the plasmogamy, and we get, again, this dikaryotic mycelium. Remember, the mycelium is just the collection of the hyphae. Notice in this picture, if you look a little closer, this must be septate hyphae, right? See the little dividings between them? So we get these individual cells. And remember, the thing about cells in fungi is that's a kind of a loose definition. In fungi, it's not about the cell in terms of reproduction. It's really about the nuclei. All right, so you got this. And notice in this organism, the basidiomycetes, the club fungi, that heterokaryotid is a lot longer. So it grows or it stays in that heterokaryotic or this dikaryotic. Heterokaryotic would be like if you were a synocytic hyphae. Remember the ones, the open tubes? Many nuclei, heterokaryotic, but there are only two, in those septate, then you're dikaryotic. So you stay that way for a long period of time. As I said before, genetic recombination, variation. And then one of the advantages, remember I told you about the hyphae, they're kind of like pipes, all connected. So when these things want to grow, They can grow really quickly. All they got to do is pump cytoplasm. It's like bulging out the ends of these hyphae, and that's why sometimes when you wake up in the morning after it rains, you kind of go, "Hey, where'd all the mushrooms come from?" Remember, they were always there. They were underground, and then when conditions were right, all that cytoplasm was pumped into the end of those dikaryotic mycelium, and blip, all these toadstools popped up with all their gills. Look at that, with huge surface area. That's why the gills are winding back and forth. Billions of spores, right? The numbers thing. And then that thing is not an ascocarp, but a basidiocarp, triggered by the environment, right? It rains, and you're like, oh, if I were a basidiomycete, if I were a toadstool, I'd say it's rained. I bet you a lot of spores would make it out there because it's nice and wet. Boom, let's all pop up. Run, fast, 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 okay? So the toadstool is a short period of time. But the actual heterokaryotic or the dikaryotic stage has been going on for quite a while. So all these toadstools pop up, make all these basidiospores, right? Fuse the nuclei, and then those are going to undergo meiosis, so you get variability, and now you got not ascospores, but basidiospores, right? So you get variability here, you get variability in mating types, and you get numbers. All right, so there was a question in your lab manual. It asked you, it said, in terms of where the spores are produced, how do ascomycetes, sac fungi, and basidiomycetes differ? You guys happen to make note of that in your lab manuals when you're looking at that under the microscope, in terms of where the spores are produced in those two different kinds of fungi. No. Yeah, inside versus outside. There you go. Excellent. In that order too, right, Dustin? So the ascomycetes are inside. Right? There's Peziza. The spores are on the inside. And then Caprinus, the other one we looked at, which is this one. You got the gills. You got the club. And the spores are produced on the outside. All right, now back to these toadstools that pop up really quick. You may have woke up one morning and see something like this in your yard. It's not always the full circle, though. Sometimes it's an arc. But notice it does grow in an arc. You guys ever seen something like, they're called fairy rings because years ago, before science was prevalent in our everyday thought, it was thought that these magical beings or whatever they're called would come down to Earth and dance around and throw, I don't know, mushroom dust or some stuff like that. And since they would dance around in a circle, that's where you get these mushrooms growing like that. I'm not sure what fairies and mushrooms had to do with each other. But now, could you help that kid understand why those things have grown in a circle, now knowing what you know about the way fungi grow? I mean, it's not a coincidence that they're in a circle, right? Who can give me a good explanation as to why, or give the kid an explanation? No? Okay. Okay. All right. Icebergs that grow out. So you're saying basically from a small little ice crystal, they keep adding more and more till they get bigger and bigger? And Harrison, you're saying that, all a mycelial mat can extend out from one spore? So if a spore landed right here and germinated, and the mycelial mat grew out, and it's growing, and it's growing, and it's doing its exozyme thing, and it's living, and everything's fine. It's all underground. You don't know it's there. And then it rains one night. The conditions are perfect for making kids. So what we're going to do is pump that cytoplasm into the tips of those hyphae, plump them up, bloop, they're going to shoot above ground, and we got ourselves a ring of toadstools. And the reason why it's in a circle is because, for the most part, when the hyphae grow out, they kind of, unless there's some difference in the environment, and that's why you sometimes don't see a full circle, the mycelia are going to grow up pretty much at an equal rate. which I'll bet you that, I was thinking about that this morning. I'll bet you that's how they can document rate of fungus growth. You know how fast mycelial, how fast hyphae are added, how fast the mycelial mat grows, right? It'd be hard to do in the wild, but if you took it into a laboratory and you put that spore into the right conditions, and then you waited and timed how long, and then gave it the right conditions to pop up with toadstools, you can measure the time and how far it spread. You know how fast they do that. It's probably a lot faster than we think. Maybe we don't want to know how fast they do that, right? Throw underground. Okay, so fungi in the environment, we've gone over all this stuff pretty in the past, right? They can break down cellulose, that stuff, lignin. Lignin, remember, is that big, really durable molecule. Durable because it was hard to break it down. So lignin was really important because, or I'm sorry, decomposers. It was, we talked about lignin first in prokaryotes because there are some prokaryotes that can do it. But they can break down that substance, which recycles all those elements that are necessary. They can act as a mutualists, right? They can act as mutualists with plants, with like we talked about in the lichens, with the algae, the blue-green algae. They can act as mutualists with animals. They can act as mutualists with animals in terms of like in the gut of a herbivore, right? It's not just bacteria that might help Bessie digest cellulose and lignin. There might be some fungal species in there that do it as well. Of course, you know, the ones we typically are going to be the parasites, the pathogens, the ones that get a bad press, right? I've showed you this table before. I don't remember, right? Have I? I'm asking, actually. I've showed you this table before, right? Yeah, okay. I just think that's a really good way to get those relationships straight. Okay, mutualistic. Let's talk about some examples of mutualistic fungi with plants. Well, first of all, mycorrhizae. We've talked about all that. And then there are these endophytic hyphae, which are hyphae or endophytes, fungi that form these endophytic hyphae. And in this particular case, the fungus or the bacteria lives inside of the plant. It's usually an ascomycete if it's a fungus. And what these things do is they're not involved in much as the mycorrhizae are in absorption, but what they can do is they can protect plants. You see, if you're a plant, if you're preyed upon by herbivores, you're kind of at the mercy of you know, the herbivore in the sense of you can't get away from it, right? Plants can't run away from the cows. Ah, here it comes, run. So they got to stay there. So one of the ways that plants can kind of dissuade herbivores from grazing on them or eating them is to have, say, maybe these mutualistic fungi that produce a toxin or a bad tasting substance. So the herbivore's like, ugh, don't eat that again, right? Those leafcutter ants, that's really cool. What they do is fungus farming. And I got a little thing on the right shows you how they do. They feed the leaves to the fungus digests the leaves, and then in turn, the fungus produces these really very nutrient-rich nuggets, or at the end of its hyphae, a little hyphal tip. So the onions. So the ants chew off the hyphae. They get the food. Isn't that neat? Fungus and the story of the fungus and the leaf cutter ants. Because look how they can chop it up with little mandibles. They do a really interesting job of a nice straight cut like that. It's amazing. Some ecologist I remember years ago said that on one tree in South America, he found something like 47 different species of ants. It's amazing. The diversity, right? 47 on one tree. They estimate more species of fish in the Amazon River than there are in all the rivers of America. Speaking of weird things, you notice I put species in quotes here because there are 15,000 species of lichens, but remember, this is really kind of pushing what a species is, right? Because like I said, this is a fungus and an algal, or a green algae or a blue-green algae live together. These are obligate; they have to live together. Very often, the fungus is a sac fungus, an ascomycete. So this is a mutualistic relationship, right? The fungus protects the algal cells from having too much light. You can sometimes have too much light. So the algal cells need enough to photosynthesize, but maybe the fungus, it's like sunglasses, right? You guys still got to see, but you don't want to be blinded. And they also prevent maybe the algal cells from drying out too. Of course, the algal cells, they photosynthesize, so that's the food that they give back. That's their part of the mutualistic. Oh, yeah, I wanted to pull this up before class, but I can find it real quick. So it's called match stick. The other day we were out walking around at Pine Lakes Preserve, and we saw, I'm going to send you a link, and we saw a lichen. It's not a deer lichen. Pull that up. What it is, it's called a matchstick or British soldier lichen. I'll show you. If you remember, or the picture you're looking at, those little cups, what happens in those little cups is these things called Cerridea. Cerridea are asexual reproduction. See what it is in the picture? It's a little chunk of fungus and a couple algal cells. This is what they look like in the matchstick lichen. Isn't that cool lichen? Neat. So that's how these things reproduce. That's what's meant by Cerridea. Now, in spite of the fact that these things look like they're kind of sensitive, they're many of the species are pioneer species. Pioneer species is the term we use to refer to. It could be a plant, an animal, fungus, could be anything. Is any kind of species that can kind of make do in a pretty austere environment, right? Kind of like the pioneers did when they moved out west. So the first organisms that moved out on the land, right? The plants and stuff. Remember how we talked about that? Those would be pioneer species. They can kind of make it, and with just a little bit. The plants had the fungus to help them, but that was a tough life when they first moved out onto land and hadn't quite adapted all completely the way that they eventually did. Now, the lichens have been pretty good at this pioneer species. We find them in the fossil record from way back 420 million years ago. And now the next thing I'm going to tell you kind of seems a little counterintuitive. Even though many lichen species are pioneer species, many of them are also what we call indicator species. Indicator species are another example of an indicator species is canary in a coal mine. You guys ever heard that expression or that saying, canary in a coal mine? Does anybody know what that refers to, if you have heard it? No? Isaac, what's canary in a coal mine? What's that mean? The Police. It's actually a line in one of the I can't remember the name of the song, but Sting and the Police. He sings about a canary in a coal mine. Right. Birds, and we'll talk about this later. Birds, because of reasons that are probably obvious. The fact that flying is pretty doggone metabolically expensive, right? Birds are really good at extracting oxygen out of air. They're much better than any other organism out there. And the reason why, and like I said, we'll look at the mechanics of this later on, is they actually, every time a bird inhales it actually takes or extracts oxygen from the same breath of air from the same inhalation more than once, right? We inhale in our lungs, we extract oxygen, CO2 gets dumped into the lungs, and we exhale. Birds are able to actually take that breath of air and absorb almost every last bit of oxygen out of it. They need that because of their high metabolic rate. So that's the good part. Unfortunately, if you breathe like that, if you're really efficient in extracting every last little chunk of nugget out of the air, if there's toxin in the air, if there's things like carbon monoxide, which collects in coal mines, or methane, which is explosive and collects in coal mines, then you're going to be the first one to feel that before anybody else does. So coal miners would bring a canary down in the coal mine, And when the methane levels got too high, well, when the bird died, they'd leave. When the bird died, they knew, oh, bird's dead. There must be something wrong with the air. We need to get out of here. Nobody quite understood why, but it was because, like I said, birds extract, and of course they're going to be more sensitive. So, indicator species would mean that the ones that are most sensitive to changes in the environment. Many times lichens are that way. Ecologists who might be wondering if ecosystems being bothered by pollution, upwind, right? Some power plant or something like that. One of the things he or she might make note of would be the lichens on the trees. A good healthy ecosystem would often have, lots of lichens, and as the environment became polluted, air polluted would be the most thing I would think about, the lichens would start to die off. So if he saw the lichens dying off, then they would say, oh my goodness, there might be something going on here, right? Canary in a coal mine. Poor canaries. I'm sure they didn't have any say in it. Says my computer is about to restart in 14 minutes. Well, if it does. You can't restart. Okay, if it does, and for some reason it says my computer is about to restart in 14 minutes. If it does that, then I'll pick back up wherever we get back on. I don't know why it's doing this. Weird. All right, let's see if we can get at least through the fungus. So lichens, I asked you in lab to identify, not in the field notebook. In the field notebook, I want you to identify down the species as best you can. But you do need to know the 3 types, and they're based basically on shape. So the crustose ones are crusty. They look like crusts of paint, right? That's what they look like. That's the red and the blue-greenish color that you see on the trees, like out on the nature trail. That's crustose. Folio means sheet. Like if you had a portfolio, right? You have many sheets of paper. So folio means sheet or leaf-like. Those are the leaf-like. And then the ones that are bushy are fruticose. I don't know why it sounds like fruit, but they're shrub-like, okay? So you need to be able to identify them from there. All right. Deer moss, of course, you're not going to put in your notebook as a moss. You won't get any credit for it at all. Here's one of the lichens we saw on the nature trail, the Baton Rouge one. Baton Rouge red stick, right? That's a crustose lichen. Xanthoparmelia, the one on the bottom, is a foliose lichen. Right? Know your lichens. Okay, so like I said, not all fungi are leave us alone. In case of, like, for example, if you're a chestnut tree, you guys probably have never seen chestnut. I don't think I've ever seen a chestnut tree, but it used to be all over the United States. But fungus can act like a parasite or a pathogen. In the case of the chestnut tree, it was an organism called the chestnut blight, brought into New York City, probably on some incoming ship. I think the organism originated in Asia. So some goods being traveled over from Asia, brought into New York City in 1910. What have we got here? 30 years later. That's the limit of what says there. 50 to 99% of the chestnuts were dead. Now most all of the chestnut trees in the United States are dead. There's a few isolated stands of them. There's an old account that said that back in the days when there were lots of chestnut trees, that a squirrel could actually walk all the way across the Appalachian Mountain Range from one chestnut tree to the other, never touch the ground. That might be a little bit of hyperbole, but still, it's kind of cool. Okay, so about a third of these are parasites or pathogens attacking plants, food crops. That's a problem. We lose about 1/10 to about 1/2 of our food crops. Or these poor people that during the Salem witch trials, because they were acting so strange, they thought they were possessed. Remember that whole, you read about that in college or in high school probably. But they think that what may have been was on the rye wheat or the rye grain they were using to make their bread, a type of fungus known as an ergot grows. And this particular that was growing on the rye actually produced a substance, it produced LSD is what it produced. So these people were eating this bread, not realizing that it was tainted with this hallucinogenic substance, and so acting strangely. You know, you can imagine back then not knowing that for sure. You think the person is possessed. This is that stuff I was talking about, the smut on corn. This is what the that word right here. That's what it's called, the delicacy, huitlacoche. Again, something I would like to try, but I never have. I haven't seen it. Okay. Oh, the amphibians. Yeah. The organism is called Batrachotrichum. Let's try that one more time. Batrachochytrium dendrobatidis. Dendrobatidis, the poison dart frogs down in South America, the brilliantly colored ones are called dendrobatid frogs. So that's where that comes from. But they suffer from this infestation of a chytrid, Chytridiomycota, a chytrid fungus that calls chytridiomycosis, and it kills like this frog that you see in the picture here. Kind of like the chestnut blight too. Look, the red is confirmed areas in this picture here. The blue, I believe, is thought to be confirmed, and the white is just a report. But look how quickly it spreads. Mexico City in '74. What, 30 years later? Yeah, absolutely. And if the decline or extinction of about 200 species, if you don't understand the significance of that, then you haven't been paying attention, right? They're all fit together. For one thing, can you imagine in Florida without all the tadpoles that would be hold on. Okay, why is it doing this? Without all the tadpoles, we'd have a lot more mosquito larvae. They feed on a lot of those mosquito larvae. Humans and animals can also suffer from things called mycosis or mycoses, which are fungal infections like Candida, which is the organism in the green picture down below. Candida can actually is a yeast, and it can cause what's called thrush vaginal infections. Females oftentimes, if they're taking an antibiotic, soon afterwards will suffer a yeast infection. And the reason why that is there are good and bad bacteria, but the good bacteria, one of the things they do is they protect your body from outside invaders. So you would imagine they would be found in areas like openings, okay, like the tear ducts, for example. Well, if you take that antibiotic and you kill off those bacteria, then opportunistic fungi like this yeast here, this Candida yeast, can begin to grow and survive, and that's where you get the infection from. That's why that happens sometimes after taking antibiotics. The other one there is the one I mentioned to you guys before, the rare form of pneumonia that AIDS patients sometimes die of. If you have a compromised immune system, then you're more prone to that, obviously. Athlete's foot, ringworm, which is not a worm. It's actually a fungus. That's what ringworm looks like. This is a section of what ringworm looks like in your skin. There's the actual fungus right there. You can see the hyphae. For the bad, there's the good, the food fungi, the ones that make alcoholic beverages and bread, cheeses, penicillin, of course. Penicillin, its benefit, we all know, is an antibiotic, but do you guys know how that came about, how that was discovered by accident? It's one of those accidental scientific discoveries. This guy was working on a bacterial experiment and had inoculated a petri dish with some bacteria, laid it in his lab, and the window was open, and over the weekend, I think, or a period of time, a fungus, Penicillium, had blown in, a spore had blown in and settled on the petri dish. And when he looked at it, he noticed the penicillin growth, and then, like in this case, the staphylococcus, but look right around it, there's no growth, inhibited growth. He's like, antibiotics, right? Of course, like I said, we can use yeast for, fermenting carbohydrates, for making beer. In genetic research, many times fungi have a relatively simple genome or genetic makeup, so we can use those in genetic engineering. We can actually use them to create things like an insulin-like growth factor, which actually would help people that have diabetes, right? Diabetics, have problems with their insulin control. Okay, folks, that's it for the fungi. Any questions on that? All right, according to what it says here, my computer is going to restart in 6 minutes. So I'll tell you what, I'm going to restart it right now. Y'all stick around. I'll be right back on in no more than just a few minutes, okay? So don't go anywhere. You don't have to sign out. I'll be right back. I don't know how that affects the recording. Let me